Thank you very much to everybody. Good morning, good afternoon, good night, depending where your time zone is. Welcome to our fourth lecture of this series organized by the International Society for Comparative Psychology. Today we have the honor to have a very important guest with us for this lecture. I'm not going to say that the other lectures are not so important, but Dr. Michael Domjan, it's an honor to have you here. It's really interesting what we're going to talk about us today. And before you start your lecture, I want to show everybody a small review of the research that Dr. Domjan does. Okay. So thank you so much, Doctor, for being here with us today. I give you now, you can share your screen with us and you can start your lecture as you wanted.
Thank you very much, and thanks for everyone who is attending today's presentation. And I would also like to thank Dr. Eduardo Reynosa Cruz for producing this series of lectures, and he's done such a marvelous job and such a great service for the International Society for Comparative Psychology. Today I'm going to talk about Pavlovian sensitization. And if you haven't heard about that before, you are not alone. No one's heard about this. I just coined the phrase, and this will be my first presentation on the topic. So Pavlovian sensitization, those two words probably seem like they're contradictory. That is, Pavlovian conditioning has to do with the learning of an association between a conditioned stimulus and an unconditioned stimulus. So the emphasis is on associative mechanisms, and all studies of Pavlovian conditioning are very careful to include control groups that rule out sensitization. So if we use control groups to rule out sensitization, how does it make sense to talk about Pavlovian sensitization? Well, I hope this puzzle will be not so puzzling. I hope to make a convincing case for the concept in the next few minutes. Or I've never given this presentation before, so I'm not quite sure how long it will last. I hope it'll be of some interest. Now, this notion of Pavlovian sensitization sort of originates actually in terms of its theoretical background with the behavior systems approach to studies of learning. And behavior systems approach, as you probably all might know, was pioneered by Bill Timberlake, and I've contributed to it substantially over the years, as has Mike Fanslow at the University of California at Los Angeles. We've all been studying behavior systems in Timberlake's case related to feeding, Fanslow related to defensive behavior, and I've been studying sexual conditioning Now, one of the core concepts in behavior systems is that a system of behavior consists of a series of response modes and modules. And each response mode or module is characterized by certain responses. Another aspect, though, of a response mode is that it represents a bias, if you will, or an increased reactivity to a restricted set of stimuli that are relevant to that response mode. And there is now evidence from fear conditioning, feeding system, sexual behavior system. There's evidence for this kind of increased responsivity to particular types of cues. And that increased reactivity can be activated by a Pavlovian conditioned stimulus, and hence my term Pavlovian sensitization. So that's a bit of a conceptual background. Let's get into the data, and I think some of this will begin to make a little more sense. So I'm going to talk about experiments, first in the defensive behavior system, then in the feeding system, and lastly in the sexual behavior system, that specifically illustrate this concept of Pavlovian sensitization. So let me share my screen, which means you no longer have to look at me. Let's see, here we go. Okay, I made this, I hope, attractive opening slide. So I said we're going to start talking about experiments. And here's the first experiment. So this is as far as I can tell, one of the earliest demonstrations of this category of phenomena that I'm labeling Pavlovian sensitization. And this experiment was done by Judson Brown at the University of Iowa, along with Kalish and Farber in 1951. Can you imagine? That was a long time ago. I was just 4 years old in 1951. And of course, all of these authors are no longer with us. And what they did was a standard fear conditioning procedure in which the conditioned stimulus was a combination of a buzzer and a light paired with shock. Now, usually when you do Pavlovian conditioning, you look for a response that develops to the conditioned stimulus. Well, that's not what these guys did. Instead of looking at the reaction to the buzzer plus light, they presented a brief loud noise during the conditioned stimulus, which referred to here as a probe stimulus. Now, this is back in 1951. This is a really fun experiment to read from the standpoint of technology, because they used a toy pistol to create this probe stimulus. So it's just a very brief loud noise. And then they looked at the startle response to that loud noise. And they measured the startle response by putting the rat in a cage on a postal scale. And when the rat flinched and jumped, he made the postal scale move, and then they figured out how much movement occurred and so forth. And they were able to measure the magnitude of the startle response. Now, what you see in this graph is this is successive days of training, and there were two groups of subjects. This is the standard Pavlovian kind of experimental design, where one group gets the CS paired with the unconditioned stimulus, and the other group gets the two events unpaired. But instead of plotting the acquisition of a new response to the conditioned stimulus, they're plotting the acquisition of the magnitude of a startle response to this toy pistol that's activated during the CS. And initially, the two groups show comparable startle responses. By the first day of training, the CSUS paired group begins to show a much higher level of startle to the pistol than the control group. Now, keep in mind, this pistol noise was never paired with shock, but nevertheless, they respond considerably to it. And there is an acquisition of this increased reactivity to the probe stimulus. Now, this phenomenon is called fear potentiated startle. And the fear is presumably conditioned to the buzzer light conditioned stimulus. And that kind of conditioned fear is conventional Pavlovian conditioning of fear. But where the sensitization occurs is once the conditioned stimulus becomes effective in eliciting fear, the animal becomes much more reactive to this startled pistol noise. And so that's why I refer to this as Pavlovian sensitization. Now, I'll give you a little variety here so you're not staring at a slide the whole time. This fear potentiated startle paradigm has become one of the major ways in which neuroscientists have been studying the mechanisms of conditioned fear. Mechanisms of conditioned fear are often studied by looking at freezing in direct reaction to the conditioned stimulus. So freezing is taken as a measure of fear. And another measure of fear is this fear potentiated startle. Michael Davis has focused his entire career on the study of fear potentiated startle and the neurobiological and pharmacological basis of fear using this kind of Pavlovian sensitization paradigm. So the next experiment I'm going to share with you is one of Michael Davis's experiments. Now, so he did his procedure was a little bit different. Here, the conditioned stimulus is a light. It ends in a half a second shock. And he trained animals with various CSUS intervals. And then during a test series, he presented the light and then presented a startle stimulus. In this case, it was a very brief, really loud noise, and looked at the magnitude of the startle response. And the startle stimulus was presented at various times during the conditioned stimulus. And the top two graphs show the results of after 5 training trials. The bottom two graphs show the results after 30 training trials. And the shock during training was scheduled at the time that's indicated by the solid line. So during training trials, the light came on and shock occurred at this point for this particular group, and then they were tested with a longer light and startle stimulus at various intervals after the onset of the light. And what you see is that the greatest fear potentiated startle, the greatest startle response occurs close to the time when the shock would have ordinarily occurred. But we're looking not at the response to the CS, we're looking at the response to this brief loud noise that elicits startle. And in this case, he used, an audio generator produce a noise burst rather than a toy pistol. So the methodology got a little more sophisticated. But the greatest magnitude of fear potentiated startle occurs at a time when the shock would have ordinarily happened. There's a little bit of a displacement early in training, as you can see in the top graphs. With 30 training trials, the time when the shock ordinarily occurred and the time when you get the greatest fear potentiated startle match perfectly. So you get more temporal specificity of this Pavlovian sensitization effect. So this is an experiment David did fairly early in his research program, 1989. And just this year, the fear potentiated startle was studied in this particular way, using the CS shock and probe stimulus being this brief loud noise. And you're looking at startle reactivity using a stable limiter kind of detection system. And fear potentiated startle was studied exactly with this methodology over probably more than 100 experiments in Davis's lab and other laboratories. And this general technique has been extended for use with monkeys and even human participants. So fear potentiated startle studied in this fashion is a well-established phenomenon. But Michael Davis focused on what is the neurobiology of this thing, and neuropharmacology, what are the neurotransmitters involved, what brain systems are involved, and so forth and so on. So he didn't really study the behavioral mechanisms, and he didn't spend much time thinking about, so why are we getting this enhanced startle response? Which is, I think, the interesting question to focus on. And my use of the term Pavlovian sensitization is intended to bring our attention to that feature of the phenomenon. And quite recently, just earlier this year, Mike Fancel and his research associates at UCLA have also gotten into the study of fear potentiated startle. Here, the conditioned stimulus is a contextual cue, so it's a place in which the animals are placed where they get a shock to condition those contextual cues. And so the shock conditioned animals he's calling them the stress group, and the control group is the no stress group. And the shock conditioned animals, in fact, show greater freezing. And as I mentioned earlier, conditioned freezing is one of the common techniques for studying conditioned fear. The term fear has come under some criticism in recent years, and so people have moved to use the word stress, but it's all the same thing. Okay, so that's conventional Pavlovian conditioning of fear. Where is the Pavlovian sensitization? Well, in this experiment, Fancel and his associates also did a test trial in which they presented a 10-second noise, okay? And this was a test trial, and the noise was presented in the context that had been conditioned for the animals in the stress group. And you're looking at, instead of a startle response, you're looking at what it calls an activity burst, which is measured through looking at displacements in a video recording from one frame to another. And the more displacements you see, the more quickly the animal is moving. And so you can calculate that as centimeters per second movement of the animal, these are rats, during the noise stimulus. And unlike the Davis or Judson Brown original experiment with a very brief loud noise, here the noise is not as loud, and it persists for 10 seconds. Nevertheless, it generates an activity burst, which then subsides as the noise stays on. So that's a more modern version of what I think we can consider to be an example of Pavlovian sensitization, because Pavlovian conditioning of fear sensitizes the animal to be much more reactive to the noise. And that's essentially what these data show. The no stress animals don't show this increased reactivity. And this increased reactivity is not directly conditioned. You don't see it when you put the animal in the box. If you put the animal in a box, you see freezing. So what the conditioned contextual cues do is they increase reactivity to this noise stimulus. Hence, my characterization of this is Pavlovian sensitization. Let's move on to the feeding system. And perhaps I should have asked Dr. Reynoso Cruz whether we should ask for questions during the presentation or just keep on going and ask for questions at the end.
As you prefer, Doctor. If somebody has a question and have some doubts, you can ask right now.
I'm happy to answer a question now, because we're finished talking about the defensive behavior system. We're going to move on to the feeding system. So this would be a good time for a break if somebody has a question.
Sure. If somebody has a question, you can ask on the chat, or you can hold your hand and I can activate your microphone.
Not seeing any hands or questions.
Yeah, looks like we don't have doubts at the moment or questions.
So we'll do the same thing. We'll take a short break after we talk about the feeding system. And so keep that in mind in case you have questions as we go along. So let's move on to the feeding system, which is shown here as conceptualized by Bill Timberlake. So I mentioned that the behavior systems approach talks about well, I just see there is a comment in chat now. No, I can't read the comment. Well, I have to move out of.
Yeah, the comment says all good for now so far.
Okay. So the behavior systems approach talks in terms of response modes. And in searching for food, there's a general search mode where you're looking all over. If you know specifically where the food might show up, your search becomes more physically focalized or restricted. And then when you actually find the food, you move into the consumatory behavior mode where you're actually chewing and swallowing the food. Now, as I mentioned, the general notion is that with these behavior systems, there's also increased reactivities to certain kinds of stimuli. And what might be those kinds of stimuli? Let's see. Okay. Oh, there we go. Okay. So this is this is an experiment that was done by Kathleen Silva and Timberlake a few years ago, in which he used two different CSUS intervals. Interestingly enough, the Davis experiments also manipulated CSUS intervals. And one group got a conditioned stimulus, which in this case was a tone, followed by food 16 seconds later. So the CSUS interval was 16 seconds. For another group, the CSUS interval was much shorter. And if you look at direct conditioned responses elicited by the tone, probably in either case, you don't see much. You don't see any direct responses. And so Silva and Timberlake introduced a probe stimulus. Now, the probe stimulus has never been paired with food. And in this case, the probe stimulus is a rolling ball bearing. So this shows a diagram of the apparatus. They put a ball bearing into the box at this point, and it rolls down this channel and exits the other side. And the question is, does the animal interact with this ball bearing? And if they were conditioned with a 4-second CSUS interval, you turn on the tone, you put in the ball bearing, and the animal pretty much ignores it. And the interpretation there is that with a 4-second CSUS interval, you're generating focal search responses, and chasing a ball bearing is irrelevant to that. Chasing a ball bearing is kind of like chasing a prey object. You know, if there's a grasshopper or something that the rat is interested in catching as food, then perhaps the ball bearing will activate that sort of chasing behavior. And that in fact happens with this 16-second CSUS interval. You notice if the ball bearing is put into the box with the tone, then the ball bearing generates quite a bit of contacts and chasing. The rat's very interested in this ball bearing. If the ball bearing is put in there by itself, the animal is much less interested. So another example of how a conditioned stimulus, in this case a tone paired with food, activates or sensitizes reactivity to certain cues, in this case the moving ball bearing. Kathleen Silva and Timberlake did a second experiment in which conditioning involved pairing or presenting food every 48 seconds. So it's what they call a 48-second fixed time schedule. But different periods during that 48 seconds were signaled by different stimuli. So these were called clock stimuli. So the stimulus number 1 was present during the first 12 seconds, then it switched to stimulus 2, then 3, then 4. And so because the clock stimuli occurred in a predictable fashion, the animal learned that, S1 occurs fairly far from when the food will actually happen. S2 is a little bit closer. S4 is very close. And if you look at nosing, the feeder during these different clock stimuli, you get the most amount of interest in the feeder during S4. And interest in the feeder is a focal search response. So S4 elicits focal search behavior. If you look at contacts with the ball bearing, they also tested these clock stimuli with the ball bearing rolling across the chamber. And this was intended to capture whether the general search mode was activated. And what's interesting is that the ball bearing generated greatest contact behavior on the part of the rats during the second clock stimulus, not the first, not the third, not the fourth. So at a certain point during this interfood interval, there was sensitization to a moving object. And that was detected and measured by a number of contacts with the ball bearing. Okay, so that's evidence of Pavlovian sensitization in the feeding system. There are far fewer experiments along these lines than we have actual data on fear-potentiated startle. But I didn't manage to find these 2 examples. There may be others, but I didn't see any others. And we could take a break here if someone has a question, and someone typed a question into chat. The comment is all good so far. Well, that's a good thing.
Any comments or questions? Well, I want to make just like a question now that you presented these two examples. Normally, when somebody teach us sensitization, it speaks us about kind of a general process that is more related with kind of fearful reactions. But now you are showing us that it's not always related with that. And in
It's
that way,
not limited. It's not
yeah,
limited to being fearful. That's right.
yeah, exactly. And then, so now you will say that it could be more a general process that could be related with any other kind of behaviors, like that could produce higher frequencies or increase any variability or parameters of the behavior. So increase some parameters of the behavior in general.
Yes, and we'll move on to talking about sexual conditioning, where there are quite a few examples of this sort of Pavlovian sensitization. So I think the phenomenon is quite broadly, quite widespread. Cindy has her hand raised.
Yeah, thank you. I don't know, is my mic working? Can you hear me?
Yes, I can hear you. Yeah.
Awesome. So I really love how you're putting this all together. Of course, I collaborated with Michael Fanzolo, and I remember Bill Timberlake presenting, so it kind of rings bells with me. But my question would be, if we're thinking of it in Pavlovian sensitization, which I love the term, I think it captures all of these phenomena really well. Would you have a prediction when the CS is maybe competing in different systems? So I'm thinking here in like Jean-Sak Kim's work, right, where he has like the robot rat or cat, right? And you have a rat that's foraging. So if you have a CS that's for the feeding system, but then you have a predator, so there's a threat coming, would you predict differences in sensitization that way? And is it related to maybe drive?
That makes the story a whole lot more complicated. And I would recommend trying to understand basic processes involved at a simpler level to begin with before we branch out. But you're absolutely right. Yeah.
I mean, I was wondering if we're seeing this as sort of like sensitization being a heightened arousal, and so there it might tap into any behavior system that's activated, right? And so
that's a great question, and we really don't know the answer to that yet. At the end of my presentation, I will list a number of issues that I think need to be seriously looked at if we want to develop this line of thinking more fully.
Thank you very much. Really enjoying it.
Absolutely. Are we ready to continue then?
We have a question on the chat now. Juan Carlos is asking, considering the food studies, what if the food produces effects on the central nervous system? For example, if alcohol were used, would the result be similar?
Well, it all has to do with alcohol as an unconditioned stimulus may activate a different sequence of response modes. And to the extent that the unconditioned stimulus activates a different sequence of response modes, the desensitization effects are going to take different form.
Thank you, Doctor. We can continue if you wanted.
Okay. So let's move on to the sexual behavior system. And I've conceptualized this system kind of parallel to the feeding system, in that if you're interested in sexual activity with a mate, you first have to find such a mate. So there's a general search mode, followed by a focal search mode. And once you actually do find the mate, then you can engage in courtship and other more intimate sexual responses. Now, we study sexual conditioning much like any other form of conditioning, where you have a CS followed by a potential sexual partner in a paired group, and the unpaired group gets these two experiences in an unpaired fashion. And we typically look at, what happens, how does the animal's behavior change in response to the conditioned stimulus. And we've used a variety of different kinds of conditioned stimuli, certainly contextual cues, which are spatially distributed, localized stimuli, and then we've also used more naturalistic, ecologically relevant cues, and we've put together a sort of a model of this diagram of, if you will, this diagram summarizes lots and lots of experiments with both rats and quail on sexual conditioning. And the stars and the squares, I think stars represent quail data, squares represent rat data. These are direct responses that are conditioned to the conditioned stimulus. They're direct conditioned responses to the CS. That's sort of the conventional Pavlovian conditioned response, or Pavlovian CR. But one of the things you notice in the summary diagram is that we've got these arrows. What are these arrows? Well, let's look at this one, for example. This arrow says we condition contextual cues, and those conditioned contextual cues change the reactivity of the subject to species typical or more ecologically relevant stimuli. So some of these, not all, some of these arrows represent what I'm calling Pavlovian sensitization. So let's look at some specific examples of that. So here is an early experiment we did, published in 1989, where we did essentially contextual conditioning, kind of like the fear conditioning Fancel experiment I showed you earlier. Animals are put in a particular place where they get a chance to copulate, and then you're interested in to what extent has that place acquired conditioned properties. And if you look at what happens when you put the animal in that place, unlike the context fear conditioning studies where you see increased freezing in the conditioned context, you put the animal in a context that was sexually conditioned and you can't tell whether the animal learned anything at all. But if you put in a probe stimulus that looks like this, and then look to see how the animal interacts with that probe stimulus, particularly in this experiment, what we looked at is, does he actually try to mate with this probe stimulus? Then you find that if the context has been conditioned, you get more grabs than if it wasn't conditioned. You get more mounting responses to this object, and you get more actual cloacal contact, those actual sexual contact responses. So the reactivity to this probe stimulus is substantially enhanced when you present the probe stimulus in this conditioned context. So that's one of our early examples of Pavlovian sensitization. This next experiment looked at, in the Davis experiments, that showed you that these effects are a function of how they manifest themselves as a function of a number of training trials. So we thought to push this to the limit and ask the question, can you get sexual context conditioning in a single conditioning trial? So we only did one conditioning trial. Again, if you look to see how the animal's behavior changes when you put them in that place, you don't see any effect. But if you place this probe stimulus into the context, the conditioned subjects are much more interested in that probe stimulus, as shown by time near the probe object, not time near the object. much more interested in the object than if the context had not been conditioned. So this Pavlovian sensitization effect in the sexual system develops very quickly. It develops in a single trial. And this has substantial adaptive significance. We'll return to the issue of adaptive significance down the road, but here's an experiment that was done pretty much like this one, or this one. We did context conditioning. And in this case, the context conditioning was carried out over 6 trials. Animals put in the context and then allowed to copulate with a female. Control group gets context exposure and the female unpaired. And then we put in this probe object, but instead of just looking at approach to the probe stimulus, we palpitated the male rats and saw how much semen they were ready to release. And for the Pavlovian context conditioning group, the volume of semen they were ready to release in response to this probe stimulus, significantly greater than for the control group. And the millions, how many sperm were in the ejaculate was also significantly greater. So this reactivity that becomes sensitized if you're in the presence of a conditioned sexual stimulus actually has reproductive consequences, and those reproductive consequences become even more obvious when we move to looking at the behavior of conditioning of females. So this is a similar kind of diagram that summarizes results of a number of experiments on sexual conditioning with females. And squares are rat data, stars are quail data, and the arrows, what you see is there are some direct conditioned responses, but there are arrows that show that reactivity to naturalistic stimuli is greatly enhanced by conditioned contextual cues or conditioned localized stimuli. And what does that mean? It means that what's a naturalistic stimulus for a female? Well, it's the presence of a male, right? That's generally a cue that she's going to become mounted. And so what's important is not how the female reacts to a light or a particular place directly. What's important is how she reacts to the presence of a male. So in this paradigm, the presence of a male is the probe stimulus. And if the female is tested in a context that had become conditioned, she is much more reactive to the presence of the male, and that shows up as longer durations of squatting and more likely to show lordosis. Now, why is that important? Because reproductive success, at least in quail, is driven entirely by the duration of squatting. This shows the percentage of percentage of eggs that are fertilized as a result of a copulatory interaction. And the highest correlation is between the percentage time that female is immobile. So that's your best predictor for the rate of fertilization. So this kind of Pavlovian sensitization is of tremendous adaptive significance. So we're kind of starting to, I've been talking too long. So let me bring this around to a little more theoretical consideration. The most popular theory of habituation and sensitization is the Groves and Thomson dual process theory, in which innocuous stimuli are assumed to activate the SR system, and that's where habituation occurs. Intense stimuli, things that we often call unconditioned stimuli, activate the state system, and that's the site of sensitization, and how much sensitization occurs can override the habituation effect that has occurred. So how do we put Pavlovian sensitization into this framework? Well, I'm not sure, but here is my suggestion, that Pavlovian conditioned stimuli will probably activate the state system and cause some level of arousal and sensitization, which then leads to a stimulus selective facilitation, which modulates how species-specific cues, sign stimuli, ecologically relevant stimuli, and other kinds of stimuli activate the SR system. So I think there may be a way. This is a first suggestion. I invite folks to think about this and develop it in greater detail. But there are a lot of things that we don't know. Lots of remaining questions. The first one is, what types of stimuli and reactions become sensitized? That is, I've talked about these probe stimuli, and in all of these experiments, there hasn't been much variation in the nature of the probe stimuli, so we don't really know what makes for an effective probe stimulus, which is the second question. So what are features of an effective probe stimulus? What is the range of sensitizing stimuli? And is this determined by the US or some other variable? We don't know that. We don't know whether the range of sensitized stimuli is determined by the CS that was used during Pavlovian conditioning. And we don't know very much about the functional significance of these Pavlovian sensitization effects, except in the sexual behavior system. There we've tied Pavlovian sensitization to fertilization rates and the number of offspring that are produced. So we know that this phenomenon directly relates to reproductive success. We know that conditioned fear has adaptive significance, but we don't know whether fear potentiated startle has adaptive. No one has, to my knowledge, has studied the adaptive significance of fear potentiated startle. And no one has looked at, in fact, there were only 2 experiments I could find on this Pavlovian sensitization thing in the feeding system. So we've got a long way to examine the functional significance of Pavlovian sensitization. And then, of course, how is Pavlovian sensitization related to other forms of sensitization? And that's the issue I tried to address in reference to the Groves and Thomson model. And finally, what's the translational significance of this thing? And I think there could be a lot of rich source of application. This is entirely speculative, and I probably shouldn't say anything because I don't know much about post-traumatic stress disorder. But what is the symptom that you sometimes see is the individual is terribly reactive and sensitive to certain kinds of cues, stimuli that were never paired with a traumatic event, that we're not present when the source of the trauma or the trauma was originally experienced. But why are you so reactive to these things? Well, it could be this Pavlovian sensitization kind of phenomenon. So, I think there's another area where I think this is highly relevant is the relationship between learning and an instinctive behavior, if you will, that is reacting to naturalistic or ecologically relevant cues. We usually think about these things as opposites or independent of one another. But if a conditioned stimulus changes reactivity to an ecologically relevant cue, all of a sudden there's much more learnable and I'd be happy to entertain any other questions people may have if I can find my curve. Stop sharing the screen. There we go. Okay, well, thanks, folks. Thanks for your attention. Sorry if I ran a little long, but there you go. We do have.
Doctor, for your amazing talk. I think a lot of us are thinking about so many things and questions that you raised with this presentation. So we have time for some questions. If somebody wants to ask something more, you can activate your microphone, or you can write or type your question in the chat. So one more question now that we can wait for the people. So now that you are showing us like that sensitization could be related with something as Pavlovian conditioning and this kind of Pavlovian sensitization, we normally teach sensitization and habituation as a kind of learning that is totally different and is not, could have some relationships with associative learning. But do you think that these, like the way we teach is like two kinds of learning that are completely separate, should be changed to like a view more like a process that could be interacted between them?
Yeah, I think the story that I tell here makes the world a lot more complicated.
Yeah, exactly.
It makes it means that conditioning effects and sensitization effects don't operate totally independently of each other. One can influence the other, and the way it does those kinds of interactions could have a significant effect on how you navigate in your world. So yeah, I think this makes the story considerably more complicated. And I'm glad there are lots of people in a group who are much younger than I am, because I'm not going to be around long enough to figure out all of this. But perhaps some of you will.
Another question that somebody wants to ask, they send you congratulations, doctor, for your excellent lecture.
Well, thank you very much. It was a pleasure. I hope I provoked some new, thinking about these things, and best of luck to all of you in your research career and other pursuits.
And here, like Laura sent you also like some congratulations. Specifically, he's saying that he's a fan of your YouTube videos. And I think many of us are waiting for new videos on your channel, doctor. It will be nice to see you again. If you can post more YouTube videos over there, will be so great.
Okay. Sounds good.
And I think it's all, doctor. Again,
Okay. Well,
the name of the International Society for Comparative Psychology. I just want to say thank you for accepting our invitation. Thank you for your time. I want to tell everybody that our next lecture will be on the 13th of October. Dr. Cindy Faz, that is here, is going to be with us, sharing her work in Apopo, ONG that works in Tanzania. So we will see what is the work that she's doing over there. So thank you very much to everybody.
My pleasure.
Hope you have a good day. Thank you, doctor, for everything.
Sure thing. Bye-bye.
See you next time. Bye.
Yeah.